So let's begin with an introduction to membranes. The first idea, or the first issue is you're a cell and there's an environment, and somehow you have to put something around yourself to be in contact with the environment. What would you put around it? Can you put something around the cell that is going to seal it such that no material gets out and nothing bad gets in? You can put lipids in, that's the answer in fact. But let's first get back to this question. Can you put a wall that lets nothing in and out to protect you completely against any bad influence? Can you do that? So lipids is the answer. This is the cell wall is made of lipids, and I'll show you a brief movie about that. Why can you not have something that is completely sealing you off the environment? Yeah?
I think it's bad for the reproduction.
That's a good point. So if you are in your city and you cannot grow, then you cannot reproduce. That's a good point. It's a very good point. Reproduction is something that gets out of the cell. Yes. But there's something even before the reproduction starts. Yeah?
Function, the cell needs things to come in and out.
So in order to function, the cell needs things to come in and out, she says. Exactly. What kind of things do you get out, and what kind of things do you get in?
Like proteins that are produced inside the cell and they get out.
So he says that proteins are produced inside the cell and they get out. That's true. Any idea what kind of proteins? Why? Why would proteins go outside? So I said everything that has to do with the function, the way a cell functions, is done through proteins. True. But everything that makes the cell function is inside the cell. Why should get any of that out?
I think in order to express the certain aspects.
You say to express certain aspects. What certain? What?
It has inherited something.
Yeah. That point is taken. You're right. We cannot reproduce, so life cannot be without sort of getting something outside of the cell. That's true. But let's just look at the point when the cell is not occupied in any way with reproduction. Why does it, for instance, need to get proteins outside? Any idea why cells get proteins outside? I'm going to show you some images of proteins that get proteins out, or that cells that get proteins outside, and why they do that and how they do that. But ultimately, yeah, that is in fact one environments. You said. Well, one aspect of environment is another cell, right? To fight with the environment, to in fact feel something about the environment. So proteins, you can think about as sensors, but also as fighting reagents, and I'll show you an example of that one. So there's all kinds of exchange, and if you want, the proteins that the cell sends out are the way a cell is communicating. So fighting is one way of communicating. But without sending proteins out, why does the cell have to bring things in and out? Or what kinds of things other than proteins does it have to get in, and what kinds of things does it have to get out? Yeah. You said the thing about in and out. Somebody else. So I'm trying to develop on your idea so that somebody else sort of pick it up. Yeah? Nutrition, exactly, energy, anything that the cell eats. So there's no cell, life is never supporting it. I do not know of any example, maybe I'm wrong here, in biology there's always an exception, but I don't know of any life form that does not take energy from outside. You cannot produce, this is like a perpetuum mobile, so you cannot produce energy from yourself. You have to get it from somewhere. So energy has to come in, nutrients, and any form of energy, right? What has to get out?
Toxics.
Exactly. Let's not talk about Botox, but some people get that in. But anything that the cell produces and no longer needs, so byproducts of what it has done, the garbage it has produced, which in fact typically are toxic for the cell. Okay, so with that, going to show you two short movies. I hope the sound will work. That briefly explain some of the aspects of the role of membrane proteins. And here it is, number one.
Lipids form a barrier that separates the inside of the cell from the outside of the cell. Phospholipids are arranged in a double layer called a lipid bilayer. A pure phospholipid bilayer would allow a very limited number of substances to pass through. Water can diffuse across because it is so small, but this is a slow process. Each phospholipid is made up of a phosphate group and two fatty acid tails. The hydrocarbon tails are non-polar, unable to bond with water, and are hydrophobic. Hydro, water, phobic, fear. The polar phospholipid head has an affinity for water, and thus is hydrophilic. When phospholipids are added to water, they form assemblies that shield their hydrophobic portions from the water. See here the formation of a lipid monolayer. The cell membrane controls traffic into and out of the cell surroundings. Like all biological membranes, the cell membrane exhibits selective permeability. That is, it allows some substances to cross it more easily than others. This ability of the cell to discriminate in its chemical exchanges with its environment is fundamental to life, and it is the cell membrane that makes this selectivity possible. In this video, you will learn about the structure of membrane and how the outermost membrane of the eukaryotic cell, the cell membrane or plasma membrane, controls the passage of substances. However, the same general principles of membrane traffic also apply to the many varieties of internal membranes that partition the eukaryotic cell. Membranes are found around the entire cell, the mitochondria, lysosome, nucleus, and many other organelles. Membranes serve to separate the contents of a cell, or organelle, from its surroundings. Here you can see a lysosome, an enzyme-containing organelle, as it is digesting an old, worn-out organelle. The membrane surrounding the digested enzymes is necessary as a barrier between the digested enzymes and the rest of the cell. The cell membrane is made up of four main components: carbohydrates, cholesterol, phospholipids, and proteins.
Okay, so let's begin with the first question. Or let's look at somehow it keeps losing the signal. I don't know why. So there are two types of cells: simple cells, bacterial cells, that essentially only have one membrane and everything else is inside. And then there are eukaryotic cells. We are eukaryotes. Eukaryotic cells have a substructure, so they have other substructures in their subcomponents that are also membrane bound. Now, you heard in this movie that, and somebody said it already, that cell walls are made up of lipids. Lipids do not let the water that surrounds a cell in. But then in this cell membrane, there are proteins, so membrane proteins. What kind of thing do you believe they do?
Could I actually ask about
Yes.
the lipids? Why is there a hydrophilic part?
So let's get back to that part. So the question, I come back to that question in a moment, to the membrane proteins, and I directly address your question now. So the question is, why in fact is there hydrophobic and hydrophilic part? So let's begin with this again. The outside typically is an aqueous environment. Inside is a cell. I said you have to think about a cell as the cells are very densely packed in a body. Right? So that means that essentially my body is almost like a solid state, although it's mostly water. And this ultimately means that the water that surrounds a cell is a relatively small part. So anyway, let's go back to this major question here. The width of the membrane is about 30 angstroms, so 3 nanometers. And the lipids have two components. They have a lipid head here, and so the major part of the lipids. Why does it have to be two different things? And that also brings up this issue, what's special about proteins in there? So certainly in the environment in which a protein typically is, it's surrounded by solvent, and this will lead to a situation in which the outside of a protein will be molecules or residues that are hydrophilic, so they want to be close to water. That will be on the outside, because on the outside you have water. While in the inside you will have molecules that want to stick to each other. They are in fact hydrophobic residues that like to group, sort of indicated by this high density of H's here. Now in the membrane, you have these lipids that have two different aspects. One aspect is the hydrophilic part of this lipid, a head group, and it has to be hydrophilic because this one actually is dissolved in water. Think about it as anchoring in water, right? While the hydrophobic inside, the lipid inside, that's the one that prevents the water from coming in. Okay, now you can sort of marvel about how come that nature has invented something that works so well for that particular purpose. We can discuss what was first here. It's a chicken egg kind of problem, but it's an ideal molecule for exactly that purpose. What you create is you create a layer of something inside here that is completely preventing anything from the outside to get in. Water. And I'm naming water because water is a very good solvent. Water is in the environment, but typically water is sort of the most dangerous thing to get in because water molecules are very small. Most other solvents are bigger, are easier to shield against. It's actually not that easy to shield against water. And this situation where you're sort of bound to water here on the end, and inside you have a lipid. Now why would you again have a lipid head group that is hydrophilic in the inside of the cell? Depending on how you see this image here, this is the lipid, say this is outside and this is inside, because inside of the cell, again, you have aqueous solution, you have water, right? So this is why, again, you have a head group here that is sort of solved in water inside the cell. And in between you create this buffer zone into which water doesn't flow. Now, let's get back to my question. Anyway, I lost the slide projector again. Why would you have proteins in the membrane? Yeah?
They can serve as gates for
They are the gates, exactly. Because what we created so far, this lipid bilayer, is something that completely shields off. In fact, if you came with a high force, you could get through, a bullet gets through, or something that moves very quickly. These bullets exist in nature. I'll show you a few examples in a moment. But this is highly, it's not easy to permeate. Now, if for your regular import and output, so the toxic or the waste products to get out, the nutrients to get in, for that exchange you need a gatekeeper, and that is exactly what proteins do. So they sit there and they provide these gates, these channels. And these channels, gates, they are in fact very specific to particular molecules. That means there is a potassium channel. And this potassium channel will only let potassium through, no other molecule. And has an amazing specificity. Now you can sort of wonder, does that mean that for every single molecule you want to get into the cell there's a particular receptor? Yes. Essentially, that's the situation. For anything that the cell wants to have, it has some receptor that reacts to that particular type of molecule. Get the projection back. Make this fast. And again, for proteins that sit in the membrane, there are two types. Those that form beta helices going through the membrane. We're not going to discuss them much because we don't have time this semester. And those that form alpha helices. And this is the example here. In this example, and the slide comes from Anna Elofsson, Stockholm University. So the blue mesh that you see here, the blue net here and here, signifies the ends of the membrane. What you see as a protein here is the one that is inside that membrane, the lipid bilayer. And as you can see, these helices are essentially orthogonal. Most of these helices are pretty much orthogonal to the membrane. They just stick through, right? In this particular case, what you see here is a 7 protein with 1, 2, 3, 4, 5, 6, 7 transmembrane helices, and these transmembrane helices, each of these essentially go through the entire membrane. Now, the particular situation energetically is, again, as I said, that typically cells are surrounded, or proteins are surrounded by solvent, by water. So you have hydrophobic residues in the inside and hydrophilic residues in the outside. In the situation in the membrane, that is reversed. Because now, on the outside, you need hydrophobic residues. You need something that is in fact want to attract to lipids. And inside you actually also have hydrophobic. So this is a different energy situation. And this different energy situation, don't know what to do about this. Maybe I have to restart. Next time I restart. Is that a question or? No. So there's one important way again that I want to make to think about this. If you put this helix through the membrane, it's completely easy to move it in that direction of this red arrow here. You can sort of move, the protein can move easily through the membrane in that direction. While in the other direction, whoops, yes, sort of indicated by this hammer here, it's very difficult to enter. So while laterally, horizontally, you can move things around, you cannot easily intrude in the membrane. And it's immediately clear why that is the case, right? Because you don't want things to intrude. And that brings up this image that I was talking about in this work from Marek Basler in Basel. And those are in fact here proteins in the membrane, a secretory system in gram-positive, gram-negative bacteria. And in fact, these things are used to shoot an arrow. So this one bacterium here builds up this big gate and shoots an arrow, which in fact this arrow is a protein, into another cell. And this is exactly the kind of thing, this is a situation of it attacks another cell by sort of shooting an arrow into that cell. In order to shoot that arrow, it builds up this mechanism, a long membrane protein with a very extended shape. It takes a long time. In fact, the length of this entire apparatus here spans half of the entire cell. And it takes quite some time to build up this mechanism, and the shoot mechanism is built up every single time anew. Now, interestingly enough, so the cell that is attacked can sense the attack. Well, ultimately it's going to die from that, but it can sense where the attack happens, and what will happen, it will actually sort of shoot back at the very same point and attack the other cell. Now imagine that one of the cells, be it the green here or the other one, is out of arrows. What do you believe they do? They give up and say, white flag, that's not what they do. Well, so far everything is pretty much like, you're smiling, but pretty much this looks very much like a sort of cartoon kind of war, right? Somebody attacks a wall, shoots in, they sort of shoot back from exactly where they shot in. But now they run out of arrows. They have no more proteins to shoot at the other side. So this is sort of the war, fire back. They ran out of ammunition. What does the cell do? What do you believe? It's a very remarkable answer. It reuses the arrows that have been shot at it. So it is so much about smiling about the suggestion white flag, right? This is pretty much exactly what you would sort of assume. So one cell is shooting arrows at another cell, proteins, and the other cell, when they run out of their own proteins to attack that cell, they take the ones that have been shot at it. Yes?
So in two different types of cells, the membrane can be attacked and is sensitive to the same kind of protein?
Yes. So the question was, if you have two different cell types here, then membranes can be sensitized or can be sensitive, can be subjected to attack. Yes, in fact they can. But But, and that is sort of the point that I try to make with this apparatus here, this is highly non-trivial. That is why you build up an apparatus that spans the half of the cell. And essentially what you get is mechanical energy to shoot the arrow so strong that it goes through the membrane. In fact, people did not believe this until Marek showed this very, very, very clearly, that you can really shoot through the membrane. This was very surprising, a very surprising finding, yes?
Is that how this bacteria works, or how bacteria infect?
This is in fact how these bacteria do it. I'm not entirely sure what you mean by is that how this bacterium works. So there are a bunch of bacteria that do this warfare. So Marek, typically biologists pick out one particular bacterium because they know how to grow it in their lab. A bunch of those.
Just of all bacteria.
We believe that essentially every bacterium, so now what people do is they essentially sequence this and see are there other bacteria that have sort of the same proteins, the same type of cells, and if so, secretion system, if so, apparatus, then you say they will all do it the same way. We have not observed that, but there's a lot of evidence. And in fact, again, so there's a prediction method, there are a few prediction methods out there that try to predict these new arrows. Okay, any other question on this issue? Yes?
So what happens if you shoot the arrow in? Does it destroy the other cell? Or?
So yes, there's a variety of reactions. So typically this is about eating another organism, which means that ultimately it's going to be eaten up, so it will die. Or it can be competition for environment. So there's a variety of things in which you can imagine that this benefits the cell.
But if it goes through the membrane, it shoots back the same protein.
So if it goes through the membrane, then the other cell should just be able with its immune system to handle that attack. Well, that depends on how much toxin or toxicity is in this arrow. So these are toxic arrows, if we follow up on that analogy. Long-term toxic arrows. Any other question? Okay, so there's another aspect about membrane proteins, and that is when you ask all the drugs that we know about on the market. I cannot remember what number was taken here. I guess in the ballpark of 1,500 different drugs that are on the market. You ask what the target of a drug typically is one particular protein. So now the question is, where is that protein? What type of protein is that? And the answer is 57% of all the targets are membrane proteins. So exactly these kind of proteins that sit in the membrane. Why? Why do you believe that's the case? Food.
Food we sell to think that it's one of its elements. For example, it's easier to get.
So if you mess with a gate, I have tons of problems today. I stumble over this thing all the time. So just a sec. Yes, you pretend that you're somebody else, and so you're messing with a gate. You pretend that you can be an intruder and you can intrude into the cell. That's one point. Yes.
Well, I think because they are gatekeepers, most of proteins on membranes are gatekeepers, so they can apply traps.
So gatekeepers, you want to get to gatekeepers. Why would you want to get to gatekeepers? So one point is because you want to, in fact, attack the cell, a cancerous cell or something like that, and you pretend that you're good, and then you're going to shoot down the cell. This is one way of doing it. What else? Yes?
You block them so that you prohibit function.
Exactly. The other one would be sort of killing, one would be blocking. Essentially, put it differently, one way of thinking about disease is disease to a cell comes from outside. There are, in fact, things that go wrong inside the cell. For instance, misfolding of proteins. That happens. Why do you believe proteins could misfold so the cell is normally doing its own life? How could it be that suddenly proteins are misfolded?
Radioactivity?
Radioactivity is one. So we have external, let's sort of make it a little bit more generalized here, rays, radioactivities, heat, something that comes from the outside. And typically, in fact, yes, it can be radioactivity, but typically the more dominant thing that happens on a typical sort of situation is heat. And a typical situation in heat is when you get fever, then in fact every single time you have a fever, some of your proteins unfold. And in each cell, there's a control mechanism against this sort of unfolding, so-called chaperones. There's a variety of things that the cell does to prevent this from happening. But if this gets too much, so if the heat goes above 42 degrees, then no chaperone can do that for the human body. And ultimately, proteins will start to unfold, and this is how we're going to die, ultimately. Yes, so by controlling what comes into, by controlling the gatekeepers, I control diseases that come in from the outside. And that is one of the major things. And that, in fact, is sort of the major explanation for the 57%, because essentially all of these drugs work against diseases that somehow are external. So the misfolding type of diseases are more rare. Okay, now, since I told you that there's no drug on the market for which any drug on the market needs a 3D structure of the protein that it targets. Otherwise, you cannot develop a drug. And that means we need to know the 3D structures of membrane proteins. Here are just a few. Again, the rods here, the helices. The layer on top, the layer below here are the lipids. In the X-ray crystallography, you don't see the lipid. So the lipid is always somehow guessed. Then you have these examples, such as this mushroom here, where you have the big protein up here, and the part that sits in the membrane sort of squeezed a little bit, those helices. A similar situation you have here. Here you have a part where you have a larger membrane proteins. I don't show it very well here, but you see a bunch of proteins. So this is what I didn't show very well before, that protein where essentially the entire protein here is in the membrane. Then you have cases such as this one, where you have a small region, something outside, cases like this one, where almost everything is in the membrane, and a lot of situations in between. Now, let's get back to the prediction problem, and let's simplify the prediction problem. So the first task of the prediction problem will in fact be that I simply say, I know there are membrane helices that go through the membrane. I want to predict where is the membrane helix and which way is the protein oriented. Now, for membrane proteins, the orientation, so does it begin on the outside of the cell or does it begin on the inside of the cell? This orientation is important for the functional type. That's important for what the protein can do as a gatekeeper. So in the first instance, we will just predict this. And for today, this is where we leave it. Now, we want to predict TMH, transmembrane helices, and the simplest thing would be to use the mechanism that we discussed so far. So something that predicts secondary structure, because membrane helices are helices. So why not just take a sequence, apply the secondary structure prediction methods that we have talked about so far, and see how well it works. And here is what you get. The red is the observed membrane helix. The blue is the completely incorrectly predicted beta strand. So proteins prediction methods tend to fail on membrane proteins. Why could that be the case? It's something that I said today. So we have a method that is trained to predict secondary structure. For proteins, it essentially I told you how we select the proteins. We take every protein we can get our hands on, and we get a method that predicts very well. And then we get such a failure. So how can that happen?
How many of the known proteins are membrane proteins?
So the question here was, I said we take every single protein for which we know the three-dimensional structure, we train our method on. The question is, how many of those proteins are actually membrane proteins? That's a very crucial question, because the answer to that is less than 1%. So it's a tiny fraction of the known 3D structures. So now comes the next point. Because there are so few in our data set, it didn't learn those. Plus, there's something else. Yes?
Maybe it's something with the solvents, because you said that the structure was different.
Exactly, it's the hydrophobicity. So it's the story that essentially the way they fold is very different. Because now we don't create a protein that is on the outside has simple hydrophilic residues. Now you need something where you sort of fold to a certain shape, but you have hydrophobic residues inside and outside. It's a very different reality. And this should somehow impact the prediction method. So that means in the simplest way in which we could do it, if I don't train a machine learning device, what would be the simplest way in which I could predict baby membrane helices? Is there anything from the introductory lecture that you remember that may give this away? What could you try to do? Yes? Just say it.
Just look for the type of, for the properties of the residues that are in the helices.
Yes, so the point is I could look for the type, so the features that make you form a helix, but that would fail, because that is what our prediction methods completely predict incorrectly. Because that's exactly what you said before, right? Because now we have a different, if you look at 99% and then you look at the property helix, you get exactly the wrong signal. But what could give you the right signal?
If you have a new protein and you want to predict, just compare with other membrane proteins.
Comparative modeling. Thank you very much. This is an important point, and it always works sort of best if there is something that you know, take all the known membrane proteins, align it, and then you sort of have one prediction. Say you don't, for that particular protein that you want to predict, have one that you know. What could you do? So, no, you don't know the function of the protein, you know a sequence. In fact, again, so historically, people, when they sequenced a protein, they knew the function. They knew something about that protein, which was the reason why they sequenced it. Today, people just sequence whatever they find somewhere in the earth, in the air, or wherever. And they don't know what the bacterium is that they sequence, and they absolutely do not know anything about the proteins they sequence. And many, many proteins remain completely unannotated. So we don't know the function. It's a sequence. And typically now saying that it has there's a membrane protein would be the first attempt to name something about its function. It's not really giving you its function at all, but you at least know something about the protein. It is in the membrane. And this first step is what we are after today. What could be the simplest way of doing that? Yeah?
Maybe the hydrophilic feature might be...
Exactly. You look at the hydrophobic feature. There's something else, remember? I told you in this image before, this image here means there is a certain width, right? If most of these helices go down orthogonal, that means that you know exactly how many hydrophobic residues you're going to have. Not exactly. This is not informatics. This is from 17 to 25 or something like that, or from 17 to something. But you know there must be a run that is at least sort of 17 residues long, and they must all be hydrophobic. In order to have a membrane helix, that's what you know. So you're going to look at what are the hydrophobic residues. I'm simply going to ask, are you going to have a run of hydrophobic residues? There is a little bit of a problem, because hydrophobicity is a very simple concept. But when you look at what I show here are three different scales. One is called GES, Eisen, Kite-Doolittle. The yellow one is what I'm going to show you data for in the next plot. But you do see in the 20 amino acids that they are not all completely agree. Okay? This is the minimal take-home message from the plot. Not all the bars are completely identical. So not all three scales are identical. There is some difference. But nevertheless, we're going to use this yellow bar, and when we do use this yellow bar, down here you see that you have a run of hydrophobic residues according to this using the yellow, the doodle hydrophobicity, and that corresponds. It's not quite giving you the membrane helix, but it sort of gives most of it. It certainly corrects for this complete bad blue mistake. Now, you could sort of refine on that, and that's a work going back to Gunnar von Heijnen. Gunnar von Heijnen said, well, we could essentially, what you're going to see here is this scale of hydrophobicity. And now you're going to look for sort of 17 consecutive that are very high, where the signal is above some threshold. But you could also sort of optimize that for something else. And that something else goes back to, there is an orientation of the protein. So orientation essentially means, does it start on the outside of the cell, or does it start, the first residue in the protein is in the inside of the cell? And what Gunnar von Heijnen realized is that when you look at the ones that are starting on the outside in blue and the ones that are starting on the inside in red, then the number of positively charged residues is different. So we're not talking about the membrane regions, we're talking about the regions that are next to the membrane helix. Okay? And when you're inside of the cell and you're next to the membrane helix, that's the red ones here. When you're outside of the cell and you're next to the membrane helix, that's the blue one. And you see that there are more positive charges in the inside. Now, if that observation is true, we could actually use that to find our hydrophobic region such that we optimize this signal. So I'm going to move my width of what I call a hydrophobic run. I said a hydrophobic run can be 17 to 25 residues. So I'm going to allow anything in between 17 and 25, such that the difference in positively charged residues between inside and outside is maximal. And that is the first method that he developed, and it works actually fairly well. Here's another idea in which you can do it. You could come up, I said there are 3 different hydrophobicity scales. Here's the idea, let's develop a different hydrophobicity scale, which essentially is a machine-learned way, if you want, to optimize prediction. So we bring up a scale that optimizes for the prediction of membrane proteins. Or, yet another idea would be to completely retrain whatever we did on the 1% of the proteins that are membrane proteins. Just take out these 1% of the proteins and redo the same that we did before. So we have some sort of local window here that does sequence to secondary structure. In this particular case is membrane, not membrane. And then there's sort of a structure to structure level. And then we're going to get a membrane prediction. And then what we get out, in fact, is 2 units. So the output here is membrane, not membrane. This is along the sequence, meaning from residue number 1 to residue number L, if the sequence length is L. That's the transmembrane unit, and that's the non-transmembrane. The signal looks pretty much like the sum over the 2 is 1. This is a neural network that trains, and it's not quite optimizing the sum to be 1, but it's actually close to 1. So what you see here is, for instance, up here you have a region that looks like a membrane helix, here's a membrane helix, and here's a membrane helix. And you already see, in fact you don't see, this is 50, 100, 150, 200 residues. So you begin to see that this, if you took the whole thing as a membrane helix, that would be an immensely long membrane helix. So membrane helices are sort of 20 residues long. This is at least 100 here, if I sort of thresholded it here. That means it's like 4 membrane helices without any interruption of hydrophobicity. And that means, in fact, we need something else. And this something else in the simplest way would be a dynamic programming-like method. Let me go a step back. Remember when I said we train a neural network, a machine learning device, the output is secondary structure, then the problem was that we got 2 short secondary structure segments predicted. And the reason for that was that in training, the way the training had to be done, we removed the correlation between adjacent residues. So the system never learned that residue I and I plus 1 are correlated. That that helices have to have at least 3 consecutive residues. The system could not learn that. We brought that into the system by coupling a second level, structure to structure level, that had the prediction from the output of the first level as input, and essentially saw that something such as HL, HL, H cannot be. And it decided either to switch this to HHHH or LLL or whatever in between, right? And this way we got a device that predicted the average length much better. For membrane helices we have the same device, but this time it overshoots. Meaning that now we really get things, so this example is very typical here, we get things where we predict 4 entire helices. And that is much too long, so we have to find a way to cut it. How would you do that? So, throwing the second level away, just taking this, the helix is too short. Putting the second level, the helix is too long. So half a second level is not possible, so you have to filter. How could you filter? Oops. Any suggestion? Yeah?
Just another question, what does the T and N
transmembrane, I'm sorry, the slides have been collected over many years and I've changed the annotation, or the way I call them. T, thank you very much for your question. T is the unit for transmembrane and N is the unit here for NOT. And what I'm showing here essentially is this output and that output unit over this sequence.
It's kind of very tough to figure out anything if you can't.
I'm sorry. No, I'm very sorry. Thank you for asking.
So transmembrane and...
Not transmembrane. So essentially, it's these 2 units, okay? We take the output value from this and the output value from this unit here, and we plot it along the sequence. I'm sorry if that was not clear. Yes?
Maybe trying to have another level.
That's a good idea. Oh, yes. I'm not entirely sure whether I never had that idea, or I didn't dare. But now that I hear the idea from you, I would be a little bit scared. Well, actually I didn't try it. That may work. I didn't try it. So, too short, too long, put a third level. And see whether the third level can learn that too long is too long. Just as much as the second level learned that too short is too short. Could be. Could be. I never tried.
So maybe the parts that are not transmembrane are going to be rightfully predicted by our prediction before.
Oh, that's an interesting idea. But it's actually going back to what he says. You mean prediction before, you mean this one? Yeah. So what she says is, if this is too short, this is too long, why, I said, this was the moment when I hit it, why not finding a 1.5, so to speak? So why not finding a way of combining them by simply saying, well, some of the breaks that this one predicts may have been exactly the ones that would cut the long helices. So find the regions in the long helices that in fact would have these breaks. This is a good idea, and this way I have not thought about it, but ultimately brings us back to what you said. This would be a great input then for the third level, right? This would be, I thought more about this way of looking at it, and thank you that you asked the question, because for this question, how to proceed, this is absolutely crucial. So this view sort of, when you look at this, does that give you some idea, some other idea? Yeah?
And maybe try a pooling filter that takes the maximum, for weights.
The pruning, what is your algorithm? It takes the maximum?
Yeah.
Yes. So we are in one of those situations. If you wrote an exam, I'm going to be nice to you, then I'm going to say you said the answer. But I'm not quite sure that I understand your answer. So what do you mean?
I mean, maximizing the likelihood that the particular residue will be at that.
How would you do that?
I'm not quite
So, okay. That's the point. That's the question. How to do that? So he says, maximize the probability. In fact, this is exactly the way to think about it. Think about this as a path. So we have the two output units, the one for not membrane and the one for transmembrane. And find the most likely path through that, if you call that an energy landscape, find the most likely path through that energy landscape. How could you do that?
Maybe again, because it seems like it's a theory.
I maybe compare.
Maybe comparing with number thing, because it seems like it's mirroring your
Oh, maybe comparing these two. No, but they are almost mirror images. That is not going to give you anything. So the only thing, so what you can, I believe you have to throw one of those away, and before doing that you normalize it to 1. So you combine them in a way you normalize it to 1. Let's assume that I had done that, then only the top, the T, the transmembrane, or the other way around. It doesn't matter, it's completely symmetric in terms of information content. But then if you have normalized them to 1, then they are no longer informative, you can throw it away. And then it's only about one of those. So then you maximize the probability for one of those. How could you do that? So you could compile a hidden Markov problem through it. It's not that trivial. But you could in principle do that. But something much simpler is this idea here. And this much simpler idea, what you argue is simpler, and it comes with a heuristic, a few decisions. So I said that membrane helix is between 17 and 25 residues. So now what you're going to do is you're going to compile all the possible segments between 15 and 25 residues that you can fit in here. Okay? Everything into that energy landscape between 17 and 25. Okay? In this example, let's assume those are all possible segments that I have. So 1, 2, 3, 4, 5, 6, 7, 8. 7. No, 8. So I have 8 possible segments. I am only looking at the ones that have in this curve here are above 1.5, because otherwise the probability will never be the highest. Okay, so where the sum over the entire segment is above 1.5. Okay, so these are my 8 segments, and in the simplest way the algorithm proceeds in the following way. It says, well, if my protein would have one transmembrane helix, which one would you pick? And by the way, the labels here on these segments essentially are the average values. Average values essentially you sum over all these points and you average by the number of points you have, residues. Right? So the maximal value is 1, obviously. So which one, if you said that my protein has one transmembrane helix, then the answer is this one. Why? Yeah?
Because the values of the T's or whatever that have the highest.
Because the 0.95 is the highest value. If you have the best bet that is compatible with your energy landscape here, the best bet is to take the best number. Right? If it's one transmembrane helix, you simply pick the one with the highest average. Because you enumerated all possible transmembrane helices. So if you have all possible in there, and if you trust your prediction, you simply take the best one. Okay? So you simply say if 1, then it's the best. Okay, now if the protein had 2, so the second highest number here is this 0.93. Cannot be that, right? It cannot be that because they overlap. If you had picked this one as a first, and that is a very good first pick, then you cannot pick this one here as second. So you have to pick one as a second that is not overlapping with your first, which would be this one here. That's the second best non-overlapping. So if it has 2, it's those 2 as your best prediction. And then you go on. If it has 3, it's those 3, and you stop essentially if the probability of continuing is lower than the probability of dropping, right? And that is as long as you add transmembrane helices. And this is not how you find the maximum, the best path, but this is how you find one very good path through it. Through the bottom. Okay? It's a way in which you can, in a dynamic programming kind of sense, very quickly find a solution to the path in which you're not going back to the first level prediction. You simply take the same energy landscape. So you take whatever you believe that since this is the best you can do in prediction, and essentially that's, again, I didn't do the third level, that could have been better, I don't know. But at this point, as far as I thought this was the best way, and going back a step I felt is not helpful, so I simply put that into an energy landscape, and if that's the best thing I have, then that's the best prediction, or the best way to sort of cut it into a prediction, yes?
Sorry, this picture is a bit confusing. Does the arrow second 0.95, does it refer to the top?
92 you mean? Oh, this 95 refers to this point. So the first one is the... Ooh. So the first one is the 95, right? That's the first yellow arrow. The second yellow arrow here is the 92. So this segment is the second highest, non-overlapping. And the third highest non-overlapping is the 89. Yes?
I don't know if this would be as quick, but if you take it as an energy landscape and look at the derivatives and the biggest differences in those, that you have a very steep slope and then a very flat curve at a high level.
And then what? In this particular case, it would give you the ends of this one, right? Which is, in fact, this is, in this way, what you would do here is, I mean, you would see the peaks, right? That's one way of doing it. And then you would sort of see the end of the mountains. You would see the mountain drops by that. But seeing the mountain drops, you would actually sort of see this long helix, which is not existing.
No, but I would not take any of that, because if they're below 0.5, then that would make the prediction.
Okay, but your eyes may be way better than mine. But you could imagine a case in which, so not assume, you can imagine a case in which this entire mountain here is about 0.5, right? And that's what you would then get with your idea. That's exactly not what you want. Because ultimately what you say in your idea is simply find my mountains. No, I don't want to do that.
Okay, but if I look at energy levels?
Yes, energy levels, I want to find the sort of different peaks. Well, okay, maybe my mountain idea here is not a good one, but you think about the slices of that mountain, right? And you essentially try different slices. And that's the way in fact, the best values will always be the peaks of the mountain. So in an energy landscape, again, so if the water rises after the ice age, then most of the mountain will be underwater, and then only some of that will be left. And that's essentially what I'm looking at. I'm sort of looking the other way around. So I cut the slice of the mountain.
How do you know how many transmembrane helices you are looking for?
So the question is, how do I know how many transmembrane helices I'm looking for? I do not know that. So the answer is I do not have any idea how many transmembrane helices I'm looking for. I'm running this algorithm until adding a transmembrane helices is not going to help in this model here. Meaning there is no, in this particular case that I show here, there's none you can add. Okay? There's none, now you could imagine that this protein would be longer, and there would be non-overlapping transmembrane helix left that you can add, because there's none left. Because say you got out of the mountain region, you cannot add a transmembrane helix here, because that is against your prediction. So ultimately, this converges the moment that adding a new unit will give you a lower energy. That's the point of conversion. It's not clear?
I don't know. I don't understand. Like, when you have like a whole area where you could find, where you found that you had a helices
Yeah.
transmembrane way.
Yeah.
How do you know how to
When to stop.
Right.
So again, my point is, so the question is, if you have this entire area here, which is 100 residues long, how do I know how to slice it? And the beauty of the algorithm here is essentially, it takes the top slice here, and it takes the top slice here. After you took these two slices, you do not have enough left and left. Some mounds will be so long that you do have a third one left. But maybe not. So ultimately the algorithm that I defined is exactly giving you the answer when that is the case or when it's not the case, depending on what you predict. And depending exactly on the shape of these mountains. So you could imagine that these mountains were shaped such that you could put more than two in there, and you could see that there are sort of two little suphills, and if you only see two little suphills, then most likely, again, depending on how steep they are, again, it depends on all the details, how many you can fit into 100. But all of that is given by the algorithm. All of that is given by the shape of the prediction. This part of the mountain looks very different from this part of the mountain. And that is because the neural network picks up a different signal in the local environment. Okay? Yeah?
I don't understand what the triangles are.
You don't understand what? The triangles? The triangles are for beauty. There's no meaning to the triangles. This is just, again, this is a plot. I'm sorry, yes, that is a question nobody has ever asked before. So the triangles have no meaning. It's essentially the energy landscape. I'm sorry. I'm sorry for that. Oh, this one's a challenging day. So simply, this is the output of the neural network for the entire protein. From residue 1 to residue N. Oh, sorry, N to residue L. This protein has 300 residues. Okay? And for all the 300 residues, I simply plot for this is a neural network that is a device that predicts whether it's a transmembrane or not for every single protein in these 300.
And how high?
The height is essentially the value here is between 0 and 1. The value here is between 0 and 1. And that is the 0 and 1 that I show for the transmembrane helix unit and for the non-transmembrane helix unit. And the curve you see is simply the curve that is the output.
That's how certain it is?
Yes. Well, that is an interpretation, sorry. Is simply the output of the neural network. You're saying how certain the neural network is about its decision is an interpretation of the situation, right? Yes. Yes?
So you're not using any information you could have on the outside parts of the transmembrane protein? Like, isn't there like a minimum distance between two?
That's an interesting one. So yes, so no, I'm not using that. So there's a minimal distance, but the minimal distance here is 1. In the algorithm, I assume the minimal distance being 1. That means they have to be separated by 1 residue. And in fact, it turns out, so initially when I first did this, I had a length of 3, but then we found things where really for 1, there's indeed only 1 residue in between. Yeah?
Maybe it's clear for everybody else, but
Thank you.
on the left side are there 3 stacks, 2 stacks, and then 1 stack?
That's thank you for these questions. So again, what exactly you have here in this stack depends on how this one looks, okay? And, obviously if you have 3 here, then you have a hill into which there are more ways of feeding 17 to 25. I'm not a good I'm not good at this. So this is not quite a one-to-one on this, right? In order to do a one-on-one on this, I would have had to have 50 different ones. You can immediately see that you can at least put 50 or more into this one here, right? But then you could never read what I'm trying to say. So this is why I had to sort of simplify and condense the reality and pretend that the hills were such. Maybe I should go back and sort of invent an energy landscape that would fit to these numbers. True, a good teacher may give me a break, guys. It took me 2 days to make this talk.
How do you get from first to the second? How do you, like first?
From the first level to the second level?
Yeah.
So from the first level to the second level, again, the way we do that, on the first level we simply, here is where the heuristic comes in. I say, from this energy landscape, I take everything above the line 0.5 that fits, that has an average above 0.5, that has at least 17 to 25 residues. I take all possible 17 to 25 numbers.
That would probably be like over 10,000.
That would be a higher number than the one I showed. And this is exactly why I'm not doing it. So this is why I should sort of retroactively re-engineer this drawing to make a different energy function. That's true. Now this is the complete set. The 10,000 or so, yeah, maybe it's 10,000. I have to think about this. No, 10,000 is a high number. If you have 300 residues, this is 7, no, it's hundreds. But still, whatever it is, okay? It's a number much larger than the one I show.
Just one which is like 50 wide, right? You could like have probably like a couple of hundreds, right? Because like between 25, you have like 7.
No, but between 17 and 25. So you have a window of 8 residues, right? You have 8, for every single one you have 8 possible.
And each one you can shift,
I would still believe it's more hundreds than...
Okay, maybe.
Because don't forget that in this particular case we have 3 membrane helixes. In this one we may have 4, but we actually may end up having only 3. So, whatever. This is a number much higher than 6. Now, I pretend that this number is 6. So this is the complete set of all possible ones. And if I had the complete set of all possible ones, going to the second level, simply the first step is, my first model is I have no transmembrane helix and I compute the probability for that. And then I have, the next model is 1 membrane helix. And I can, if I have 1 membrane helix, I take the one that has the highest value, which is this. Put it down. Now, if my protein has 1 transmembrane helix, it's that one, and I can compute the probability, given my energy landscape, that it has 1. If it has 2, and so forth, and so on. And again, the algorithm simply ends because the probability goes down. Yes?
I'm having trouble to understand this, so can you please explain it again? I'm not able to understand this. What exactly that involves?
Is anybody else having a problem? Okay. I'm sorry. I'm very sorry. Assume that we cut out the top graph, the energy landscape. Those are values. This is the first residue, the last residue in the protein, and this is simply the output of probability to be in a membrane helix. Is that clear to everybody? So far, so good. So now what I'm going to do, I know membrane helices are between 17 and 25 residues long. So I'm going to take out here say the top peak. Say the way this looks here is the top peak. So I'm going to take and say this is sort of the line that is above 0.5. So I'm going to take the first 17, 18, 19, 20, and so forth, right? So I build up all possible segments. and then I move it by one residue on, and I create all possible segments in this landscape. And for every possible landscape, every one that is between, every segment between 17 and 25 residues. 225. I build them all. For all of those, I compute the probability. Yes?
So a segment is just one line?
Yes. A segment is just one line.
And all residues on that?
Yes. Because that essentially is, we cut out 1, 2, 3, 4, 5. In this particular case here, again, this is 150 to 200. So from here to here, there's 25 residues, right? So we're looking at a consecutive stretch that is going to be a membrane helix. So I've cut out all the possible segments. Are we clear? Is everybody clear on that point? In my next slide, again, you already see here, this is going to be many. We had the discussion, is it going to be 10,000? It is several hundreds, thousand, maybe even 10,000. It's a large number. Here I pretend that this large number is 6 for simplicity. So I say on this slide, all possible segments between 17 and 25. Of all these possible segments in my energy landscape, I found 8 segments. And that is on the top panel. The 8 segments. 1, 2, 3, 4, 5, 6, 7, 8. Okay? Each of these is a possible membrane helix. Yeah?
Wait, the best segments, how do you measure which one is the best? Like,
so how would you measure which one is the best segment?
Well, probably take like a segment which contains particles with the highest probability.
Exactly. The best segment would be the one with the highest peak, right? And the highest peak essentially is the value here. The best segment is 0.95. In fact, this is typically one of those peaks here. Okay, that's the highest. And of all of these 8 segments, the top one is in fact this 0.95. So if you predict that your membrane protein, that you have a membrane protein that has membrane helices, and it has at least one membrane helix, then your best guess is this is the one you're most certain about. If the protein has one transmembrane helix, it's that one. That is clear. Right? Yeah?
Maybe you should write down that it's like a greedy first best algorithm. Like it's just taking the best and finds the best, because it's not like,
that's true.
It's not like the 0.93 one, right, on the left part.
It's a greedy.
But it's like, oh no, it's already like occupied by the 0.5 one. Okay, just.
Very true. So you make a point that I should have emphasized more. This is by no means going to find the best optimal solution. This is very greedy. You pick as long as you have one transmembrane helix, this is absolutely clear. This is the best pick. But the moment you pick 2, there is sort of some payoff, right? And this payoff, ultimately, you immediately see why I'm doing it in a greedy way, because of CPU time. There was another point that...
Why do you start from the left? You would start from the right and
That's unfair. So, but most likely, somewhere Freud put into me that I put the way I created the values, most likely, I start from the left simply because that is the highest value, not because I write from left to right. Coincidence. Data is it this way. Okay, I made up these numbers. Most likely I started with the highest from the left because I write left to
It would be more obvious, obviously, if it wouldn't be that way, right? This way you could like, what's the assume that it's from the left to right, but it's not.
Yeah, so I should change. Okay.
Maybe you start from the right and you get 4.
No, but in this particular case, it's clear why you start from the left, right? You pick,
0.95 is the highest.
Exactly. So again, the first one you pick is exactly the best choice. And that happens to be on the left. Why is it on the left? Happenstance. Yes?
If you just take the average score, it kind of, doesn't it be biased against like short sequences?
No. I mean, if you take the sum, then that would be against short ones. But the average shouldn't be against short ones. If anyway, it should be against large ones.
Yeah, exactly. Against large ones, it should like bias for.
Which bias toward. Sorry, I misunderstood what he said. That is true. The probability of a short one to make it is high. That's true. In particular, so again, it depends a little bit on how the energy landscape looks. But you're right.
In the matrix cases, yes.
But there's a tendency to sort of favor shorter ones. That's true. Yes?
Just have one more question about the landscape. So each dot is that one residue?
Yes. Well, dot is the wrong word. Imagine this is a line that doesn't have any dots. But yes, every single point, every point is one residue.
So if I take a segment, in this specific case, it would be not that many residues.
17 to 25. I take all possible segments between 17 and 25.
Like the way I'm imagining it is you take a line,
Yes.
and you see how many points.
I take all possible lines that are between. I take all possible lines of 17. I take all possible lines of 18. I take all possible lines and so forth. I take all possible lines between 17 and 25.
Is that here anywhere the case?
Is that
the case anywhere here? If you take a line here?
again? I'm not showing any line here. This is what I sort of tried to doodle here, and this is clearly not what I'm showing on the next. No, I'm not battery. This is clearly not what I'm. This is clearly not what I'm showing here. This is clearly fewer than 10,000. Yes. But in principle, that's what it is. Okay? No, you don't understand it, or you're not appreciating my article?
One line would have to be 17, then I'm imagining a lot more ups and downs.
Oh, be careful. The number of ups and downs. Again, what you're looking at here is the output from this prediction method.
Yeah.
Whether you see ups and downs or not is decided by that prediction method.
I get that.
Yes?
Every point that crosses this line, which is the segment, and that's counted as 1 and you're counting towards 17 to 25. When this curve is across that line,
I'm not entirely sure that I understand what you say, and I'm not entirely sure that you understand what I say. So let's begin with the first point that is above 1.5. For the first point, you're going to make a line of 17, and then you're going to make a line of 18, and then you're going to make a line of 19. So you're going to make all the possible lines between 17 and 25. And then you go to the second point. Maybe the second point is here. And you're going to do the same thing.
So this clear, I was thinking of a completely different direction.
Are you with me now? Did anybody manage to get off the train again? Are we still all on board? I'm going to sink this boat soon. Now, again, this is greedy. I do have enough battery left for the pointer, I hope. Yeah. So this is greedy. These are all possible segments between 17 and 25. This particular case, there's 8 of these. The value given is the average over these 8. And there are a couple of things that have been observed that are absolutely true. That using the average is sort of favoring shorter ones. Simply the way energy landscape looks, it's more likely that the shorter one gets a higher average. And it's greedy, because I say that if I have 1, it takes the top. That's clear. But if I go to the second one, just a second, if I go to the second, so if my model has 2 transmembrane helices, I say I stay away from... That's a challenge there. I stay away from, I cannot take any of these because there is already a transmembrane helix. The second transmembrane helix cannot overlap with the first one. Although I allow separation of back, I do need only separation of one. I do need a separation of one. They cannot overlap. And the second best, therefore, would be this one here. And it is greedy in the sense that the moment I get to 2 transmembrane helices, it is not compiling the best possible way for 2. So the prediction of 2 transmembrane helices could be different. Right? I could open that can and recompute for every of these assumptions, again, what would be the best model. I'm going very greedy about it. I just take the best path through that model. Anybody, any doubts? Yes?
Could you please explain once again why we have 3, then 2, then 1?
No, so first of all,
And the second.
yes, you mean on the first level?
No.
On the second? Okay. We get to the model here with 1 transmembrane helix is the best one and everybody's okay with that. Now, say I want to add 2 transmembrane helices. I will take the one that I have from the first. That's why I inherited this one here. This stays on the same place. Because I'm not going to move on my previous decision. That's why this algorithm is greedy. But I'm not going to revise my decision. This is why on this level here, for predicting 2 transmembrane helices, the first of the 2, which again, whether it's left or right, it's happenstance whether it's on the left or the right. When I say first, it's simply which is the best. Maybe I use your word best. The top of the best. So the best is going to be always the best. The second best is this. If I have 2, I take the best and the second best. Okay? And they happen to be left, so number 1 and 2 in this drawing. If I predict that there are 3 transmembrane helices, I take the best, the second best, and the third best. The best, the second best, and the third best. So you simply have 3 here only because you inherit the best. It's always the best is the best.
So is the best on the right or the left?
Forget about left and right.
What is the rectangle?
The red angle is a transmembrane helix. Oops.
So when there are 3 of them,
There are not 3 of them. I'm sorry. So the way to think about this is if my model has no transmembrane helix, if I have 1, if I have 2, if I have 3. Those are in fact 1, 2, 3, 4 different predictions. And this, in fact, happens to be the final prediction because the algorithm converged, and using 4 is not better and using 3 is better than 2. Yes?